Hello, good afternoon all. I hope you are doing well. So welcome back to the botany session. So today, yesterday we have discussed it in the cell unit of life, prokaryotic cell we have discussed. So in the prokaryotic cell, so we completed the cell surface appendages like flagella, pili and fimbriae. So yesterday we have discussed about the mesosomes. Those are nothing but the enfoldings of prokaryotic cell. The enfoldings of prokaryotic cell towards the cytoplasm is nothing but mesosomes. So, or we can call the extension of cell membrane also we can call. So, these mesosomes functions also we have discussed. So, there in your book they have given three: vesicles, tubules and lamellae. And the function of it, that is cell wall formation and increasing in the surface area of cell membrane, and enzymatic secretion, enzymatic content and secretion, DNA daughter cells, DNA duplication and distribution in the daughter cells. So, and it also works as mini power station, I said yesterday. So, it is about the mesosome. And the cell surface appendages outside. So, yesterday we have discussed the flagella, pilus or pili and fimbriae. So, flagella, it has three parts, that is basal body, hook and filament like structure, that is filamentous. So three parts are there and it is used in motility purpose, movement, showing the movement. So when it rotates the flagella, the bacteria will be moving like this. And if you see the pilus or pili, it is also made up of some special protein, that is pilin protein is used for the formation of that pili protein or pilus, the cell surface appendages. So it is used in attaching or adhering to the host tissue or rocks in the stream. So even it is applies to the same fimbriae also. Fimbriae is also same. The difference between fimbriae and pili are many are there. So we have discussed yesterday too. That is length of the pilus. It is comparative to the fimbriae. It is bit large. The length of the pilus is big there. If you see the fimbriae, it is a Bristol shaped structure, which is length of that it will be comparatively less. And pilus, it is also used in the conjugation, where a kind of sexual reproduction in bacteria, which results in variation there. So there, the pilus is used as a medium of transformation of the DNA from one bacteria strain to another bacterial strain. So yesterday we have discussed the cell surface appendages also. And coming to this ribosomes and nuclear inclusion bodies. So we have started ribosomes there. So in case of ribosomes, so here ribosomes are no membrane bound organelles. Both are no membrane. So both the organelles or both the things, they don't have any membrane there. No membrane in both. Coming to this ribosome. So there is some information which is given in eukaryotic cell concept also under the subheading of ribosomes. But now we are going to discuss only of prokaryotic ribosomes. So here ribosomes, they measure around 15 to 20 nanometer. So nanometer means 10 to the power of minus 9 that meter. 15 to 20 nanometer it will be the size of ribosome. And here the ribosome, it is made up of two subunits. One is, it is made up of two subunits, smaller subunit and larger subunit. So it is made up of two subunits, that is one is smaller subunit and larger subunit then. It is made up of two subunits, one is smaller subunit and another one is larger subunit. So actually what it composes of means, so this ribosome, it is composed of two things there. One is rRNA. So it is a type of ribosomal RNA. R means ribosomal there. Ribosomal RNA plus some proteins will be involved in it. So some proteins will be involved in it. So that together it is making smaller unit and larger subunit there. So smaller subunit, it is made up of smaller subunit and larger subunit. So it is the larger subunit here and the smaller subunit here. And even if it is, if you are taking a cell that larger and smaller subunit, density also it differs. So based on the density of smaller subunit and larger subunit, so the density here is so here in case of this smaller subunit density is 30s and the larger subunit is 50s. So 30 plus 50 when they are together. So when they are together, so the unit will be of 80s. Wait, 70s. So here some might like you might be confused here. You might be surprised that I am saying smaller subunit is 30s and larger subunit is 50s. So then when it is together, it is 70s and not 80s there. So it is even based on this, they are in Neet, they asked 3 times, 3 to 4 times they asked based on the gradient of this one. So if you see here 30s and 50s here, S means Swedberg unit. Here S word is there, no? S is Swedberg unit. S means Swedberg unit. Swedberg is the scientist. So he first observed this gradients in case of ribosomes there. The smaller subunit it has 30 years and the larger subunit is 50 years. But when they are togetherly measured, it is showing 70 years there. How in the sense, and what is this 30 years and 50 years? Whether they are having like, are they weight or any volume or any gaseous substance? No, no, it's not like that. So 50s and 30s are the density of the ribosomal of smaller and larger subunit there. So how the density will be measured in the sense? So we will be taking a test tube here, a test tube with a smaller subunit. Smaller subunit in the sense it will show the smaller subunit is showing 30 here. So when you add, so say think that there are two ribosomes packets are there. One is smaller subunit and one is larger subunit. So I am taking the smaller subunit and I am adding into this smaller subunit constituent is there, no? I am adding into this test tube. So when I am adding into the test tube, I will keep it for a centrifugation. Centrifugation is nothing but there will be a machine will be there. Centrifuge machine will be there like this, surrounded by many holes. Here you need to keep the test tubes like this. So when you keep the test tubes like this, after adding this one, when I am keeping the test tube like this, and this centrifuge machine is there, no? This much of big it will be, and I am covering this one now. It will be rotating. This centrifuge machine, it will show movements like rotation. So when it rotates, think that, see, I am adding some water and I am adding some ribosomal smaller subunit there. So when it is rotating like this based on centrifugal force, so when it rotates like this, the movement of molecules will be going down based on the density of that one. So here the density of this smaller subunit it is showing 30 subunit, 30 s there. So the movement it is showing it is 30 s there. So when you are measuring this larger subunit, it is showing 50 s. So let me draw that on. So when we are measuring this larger subunit here, it is showing 50 years. It is showing 50 years there. But when you are adding together, so together smaller plus larger subunit, when it is centrifugated, the movement of the molecules based on the density, it is showing there 70 years, not 80 years. So when both they are together, hybrid this, so we are getting 70 years, not 80 years more. So this is 50 years. Okay, I will take this one only. 30 years it is. So this is the centrifugation machine. The centrifugation machine is there, no? It will be using for centrifugation, and it works on the based on centrifugal force, and based on the density gradient, the movement of molecules will be taking place. So here it is 30 s smaller subunit is there, and here it is larger subunit which is showing 50 s. So it is showing 50 s here. So when you are measuring together, we are getting 70 s here. 70 s. It is the density gradient. So this is smaller subunit here, which is showing 30 s, and it is 50 s. But together it is there, no? Together it will be 70 s. So together it will be 70 s here. It is the centrifugation gradient. So it is in case of ribosome, it is showing this much variation. Especially of prokaryotic ribosome, it will be 70 s. If you see in eukaryotic cell, if I am taking my part of cell, and in that ribosomes will be there. So in that it is made up of smaller subunit and larger subunit. So when you take out the smaller subunit and add it in the test tube and keep it for centrifugation, the smaller subunit in case of eukaryotic cell, it will be showing 40 s. Smaller subunit it is showing 40 s, and larger subunit it will be showing 60 s, and together it will be showing together. See, larger subunit in case of eukaryotic cell, our cells or plant cells I am talking about. This is in prokaryotic cell, 70 s. See, you need to remember this one. Even last year also they asked this question based on this gradient of this ribosomal larger unit and smaller subunit there. So if you see in every CET paper or Neet paper, this question will be repeated many times. So it is very important to remember here. 50 plus 30 s, 70 s, it is in prokaryotic cell. 40 s plus 60 s, it will be 100 s, but not 100 it will be, but it is 80 s. So here our even number is there, 4 is even, 6 is even, 8 is even, 3 is odd, 5 is odd, 7 is odd. You can remember like this. So even number it will be together, 40 plus 60 it is 100, 50 plus 30 it is not 80 but it is 70 here. So it is in case of eukaryotic cell. In case of eukaryotic cell, it will be 80 s here. It is in case of prokaryotic cell. It is in case of prokaryotic cell. Not only prokaryotic cell here, not only prokaryotic cell 70 s will be there. There are some ribosomes which are present in mitochondria. So there are some ribosomes which are present in mitochondria also. They will be showing 70 s. And there are some ribosomes which are present in chloroplast. Those are organelles which we will be seeing in eukaryotic cell. So in that also it will be 70 years. So prokaryotic cell and even in mitochondria and even in chloroplast. So the ribosome, it will be measuring 70 years and even it will be having same, so 30 years plus 50 years same it will be. Eukaryotic cell, it is of this one. So you should remember this calculation and the gradient differences here. This is in case of prokaryotic cell is of this one. And there is one more special thing in case of prokaryotic cell is there. So let me erase this one. Okay, we'll see that. In ribosomes, it is about the gradient variation. And there is a one structure will be forming in prokaryotic cell, which is meant to only in, which we will be seeing in only prokaryotic cell. So you need to remember that also. That is the one more important topic. So that is polysome. That is polysome. So here polysome will be, polysome is nothing but, so it has two terms there. Poly means many and zome means ribosomes. So many, this kind of ribosomes, it will be attached to the mRNA there. So there will be an mRNA. mRNA is nothing but ribose nucleic acid. So that you will be studying in next sessions. But you just remember this one. It is RNA, mRNA it is. And to this mRNA, there will be many, this ribosomes will be attached. So many ribosomes will be attached to this mRNA. So many ribosomes will be attached. So when many ribosomes are attached, this condition we are calling this as polysome. Many, poly means many and here zome means ribosomes. So many ribosomes attached to a single RNA or mRNA, we call this as polysome. Polysome structure it is. So in molecular biology we will be learning. So there is a DNA is there. So DNA is there. DNA will be having some information, it will be storing some information. So DNA, it is like a book. So it will be having many pages of information. So if you want to read one information, one page of information, that will be copied, photocopied or transcribed in the form of RNA, that we call it as transcription. So that RNA is nothing but this strand, RNA. And RNA, when it translate that language of DNA, we will get a protein there. So we will get the protein formation there. So this protein is nothing but, protein can be anything. Protein can be enzyme, protein can be hormone, protein can be receptor, protein can be transporter. So it can be anything. So to expression of this protein, it has to follow this path. So DNA is like a book which has many pages of information. So now RNA is a one page of that information. It has to be translated into a protein there. Protein is nothing but, it is present in everywhere, like diversified form it will be there in the our body cells also, even in prokaryotic body cells also. So here in this protein, to form this one, to translate the language of RNA into protein, so it will be, it has to attach to this ribosomes, because we know that these ribosomes are nothing but what? Ribosomes are protein factories. Ribosomes are nothing but protein factories. So here protein formation will be taking place. So here a chain of protein formation will be coming out like this. So these are proteins. So these are proteins here. So these are ribosomes. These are ribosomes, and this is RNA. So RNA, when it is reading, this ribosome is reading the RNA page information and it is making proteins there. So many proteins will be found, but all proteins are similar here. Listen here, there is one page of information is there, and one page of information it is read by 3 ribosomes. So when it is reading done by the 3 ribosomes, the information will be same there. So when the information is same, the protein, the formation of protein, it will be same there. So that's why, so this polysome, it is this, in this the polysome is there, no? This polysome, it has the many ribosomes are attached to this one, so that we call it as polysome. So when the polysome is attached with this many ribosomes, it will be reading the same RNA strand, and when it is reading the same RNA strand, we will be getting the same protein there. So this is about the ribosome. And in this polysome, or it is, you can also call this as polyribosome. It is only seen in prokaryotic cell, not in eukaryotic cell. So this will be seen in case of prokaryotic cell there. It is about the ribosomes. Now coming to the inclusion bodies. So I will take another slide here. So here inclusion bodies. So here inclusion bodies are nothing but, these are also no membrane bound organelles. So here also these are also no membrane bound organelles. Here these are also no membrane bound organelles. They don't have any membrane. Then how they will be found in the prokaryotic cell, in the sense, see, if it is a cell, if I am drawing, this is a, think that it is a prokaryotic cell. A prokaryotic cell is there in the cell free system. So in this, the inclusion bodies, it will be appearing like this. They will be gathering at one place and they will be forming a, like this kind of structure. So this, they didn't cover any membrane or layer upon that. They gathered at one place in the cell free system, and it is, these are nothing but food reserve materials, or not only food reserve, even it is just a reserve material of something, some molecules gathered at one place and making a group there. So that is inclusion bodies, we call this as. So it is one here. So like this you will find many, you will find many things which are attached there, making inclusion bodies of different things, different composition. There are different types you will be seeing here. So the gathering of that or grouping of that that kind of molecules at one place. And at the center, what will be there? A genetic material. And even plasmids are seen here, small circular plasmids. And the cell membrane extends, it will be like a flagella, and it is having many pili structure. So it is the prokaryotic cell. Now what we are discussing is inclusion bodies. So the which don't have any membrane there. So now we'll discuss one by one. So the inclusion bodies are what we are seeing here. You will be seeing a different kind of inclusion bodies. So like phosphate granules is one type of inclusion body. So all the phosphate granules are accumulated at one place in a cell-free system without having any membrane, that we call it as phosphate granules. In somewhere you will be seeing like glycogen granules. These are glycogen granules, where it is like storing the glucose kind of structures, and they are accumulated at one place in a cell-free system without a membrane, that we call it as glycogen granules. And in some we will be seeing synophysian granules. Synophysian granules. So we need to discuss one by one now. So in some you will be seeing gas vacuoles also, where it will be completely, there is no space here. Okay, I will write separately. So coming to this phosphate granules, here phosphate granules is nothing but it is containing phosphate. So it is a phosphate storage body. So here inclusion bodies are nothing but what? It is no membrane bound thing or organelle, and these are reserve materials. Reserve materials present in cell-free system. So that is inclusion bodies. And phosphate storage, it will be there. See, in us mitochondria is there, the production of ATP will be taking place, and the ATP, to create ATPs, inorganic phosphate is required there. So one phosphate they will take and make ATP there, adenosine triphosphate. So here phosphate storage granules are nothing but where the phosphate will be there. So if bacteria is required ATP, so ATP is nothing but what? ATP is nothing but for the ATP is nothing but adenosine triphosphate. So how it is formed? Adenosine diphosphate plus one phosphate. So it is ATP synthesis. For ATP formation also, these phosphate granules are used there for ATP formation. There the phosphate granules are used in case of bacteria, and even the formation of DNA or formation of RNA, there also these phosphate granules are used. And coming to this granules, glycogen granules. Glycogen granules are nothing but these are storage of glucose. So glucose will be stored in case of this one glycogen granules. And where it will be used? If bacteria is undergoing starvation, think that there was a bacteria which it was present in the kitchen of your house, and it will be enjoying the food there, like lot of food is there, and it will be roaming, it will be engulfing the bacteria, the food which is present there, and it will be taking and it is enjoying. So suddenly one day, it's bad luck, the bacteria we throwed in the dustbin. There also lot of food is there in the dustbin also, because the bacteria can survive anywhere, everywhere it will. And it is shifted to somewhere like bare rock or sand, where nothing, no food is available there. So in such case what happens? The bacteria don't have food, but they require energy now to survive there. So this glycogen granules, which is storage of glucose which is present in this bacteria, it will be used under the condition of starvation. So when starvation of bacteria was taking place, when bacteria is undergoing starvation, so then there it is used as glycogen granules are used as food substances there. So that is the nothing but the storage of glucose there, that is we call it as glycogen granules. So coming to the synophysian granules. So here synophysian granules are nothing but which we will be seeing in case of cyanobacteria. So cyanobacteria are nothing but cyano, blue green algae, And where do we see these cyanobacteria? So one analogy or one example you can take in case of paddy fields or rice fields. The farmers, they will be, if they are growing rice in that, they will, what they will be doing. So if they are growing rice, so they will be making, so the water will be kept there, means the stagnant water will be remained in the crop or that field area. This water, upon this there will be, you will be seeing a green layer. So there will be seen a green layer, which the blue green algae will be there, the same cyanobacteria there. So it helps in nitrogen fixation there, means these are, if they are helping the nitrogen fixation, they should be present, they should carry the nitrogen, no? So here nitrogen will be stored. Nitrogen storage house it is. So example, in case of paddy fields, you can take this one. Examples for the cyanobacteria are Anabina, Nostex. So those are example of it that we will learn in biological classification chapter. So it is about the inclusion bodies, and one more is left, that is gas vacuoles. So one more is there, that is gas vacuoles. So these gas vacuoles also, they don't have any membrane there. These gas vacuoles we will be seeing in case of purple bacteria and green sulfur bacteria. Sulfur bacteria or green bacteria. So there we will be seeing green sulfur bacteria, we will be seeing gas vacuoles. So as its name indicates, gas means a vacuole will be there where the gas will be filled within that. So think that a bacteria which is living, think that a bacteria is living in, a bacteria is living where the water surface is, water is there. So now this bacteria requires food, and this bacteria requires food now. So that bacteria will be coming upon this layer. So the bacteria will be coming upon this layer on this surface like this. On the surface they will be floating. So if they are on the surface, they are floating, the green sulfur bacteria or purple bacteria, they require sunlight also, they require water, surface area, and some gaseous contents is also required for them. So what the bacteria is, why they are floating on the water surface, what is making them to float or sink on this water surface is, that is about these gas vacuoles. So these gas vacuoles helps in buoyancy or floating of this bacteria. Floating of bacteria, it is done by these gas vacuoles. So it is about the inclusion bodies of prokaryotic cells. So what we have learned? So ribosomes. So in case of ribosomes, it is made up of rRNA, ribosomal RNA plus proteins. So the ribosome will be having made up of two units there, smaller subunit and the larger subunit. So larger subunit, it will be, in case of prokaryotic cell, it is 50S, smaller subunit is 30S. 30 plus 50, it is 70S. This is the sedimentation coefficient we will be seeing in case of when we do centrifugation here. This is centrifugation machine. Centrifugation machine it is. So when you keep that test tube here, it will be showing gradients there. So it will be rotating in some 1000 rpm and 2000 4000 rpm. So based on that, the gradients, it will be showing the movement. So it is about the prokaryotic cell, mitochondria and chloroplastal ribosomes. This is the ribosomal sedimentation coefficient. In case of this eukaryotic cell, 40S, 60S, 80S, it is just that. So in case of the prokaryotic cell, it is specifically, it is only in a prokaryotic cell. Only in prokaryotic cell is seen. So polyribosome or polysome, where to the RNA, many ribosomes will be attached. So we know that ribosomes are protein factories. So when ribosome is reading this information, so it will make the protein. So all the proteins will be same, and this complex is there. No, the complex of many ribosome plus RNA, we call this as polysome. Polysome is nothing but the complex of many ribosome attaching to the single RNA, single mRNA, that we are calling it as polysome, and it will be producing the same proteins there. And in inclusion bodies, we have seen phosphate granules, glycogen granules, cyanophosphine granules. So according to that, different functions it will be performing by the bacteria there. And gas vacuoles, it will be helping in the floating of buoyancy there. So next we will be seeing eukaryotic cell. So far we have completed the prokaryotic cell. Now prokaryotic cell, prokaryotic is nothing but pro means primitive carrion nucleus, primitive nucleus it will be, means earlier, before when earth was originated, so only prokaryotic organisms were at that time. So as the evolutionary line, as the time passes, decades, years, million of years, so as it passes, the eukaryotic organism arises there with the um, help of endosymbiosis. Endosymbiotic is nothing but engulfing something and making a complete organelle and so cell, complete cell, or a eukaryotic cell. Eukaryotic cell is nothing but U means what here? U means here U means true, and carrion means nucleus. So it is a true nucleus is seen in case of these cells. So where do we see these eukaryotic cells then? So these eukaryotic cells are seen in case of protist. Protista is nothing but, don't think much about this protista there. Protista is nothing but unicellular eukaryotic organism. Unicellular means what? Single cell. Single cell eukaryotic organisms are placed under the group of protista. Protista is just one group where it includes organisms which has single cell eukaryotic cell it will be. There is the previous section what we have discussed, prokaryotic cell is there, no? That prokaryotic cell is seen in case of bacteria, Monera kingdom. This is Protista kingdom. So eukaryotic cell we will be seeing Protista there, and not only Protista, even some animals, plants, and some fungi, not some, all fungi. So protista, animals, plants, and fungi. So all these organisms are made up of eukaryotic cells. So you can take example of yourself, myself. So all my cells are eukaryotic cell there. Means proper. Eukaryotic is nothing but a proper, well organized cell is there. We have discussed in the an overview of the cell we have discussed, no? There you can go and check what is eukaryotic cell, an overview of the cell. So here in eukaryotic cell, so not all eukaryotic cells are same there. So eukaryotic cells are different in different organisms, and within the same organism, different eukaryotic cells will be there. You just take example of nerve cell, RBC cell, muscle cell. So all these are different, so different in appearance, different in structure, different in composition, isn't it? So it shows that eukaryotic cells are not identical, not similar. So there is a statement in NCERT where you will be seeing not all eukaryotic cells are identical or same. So they are different. And when we are discussing that protista, animals, plantae, and fungi, or when we are discussing eukaryotic cell compared to the prokaryotic cell, what makes eukaryotic cell different there? So here eukaryotic cell, it will be having some locomotory structure also, locomotory or cytoskeleton structures are seen in case of eukaryotic cell. So what are those locomotory or cytoskeleton structures in the sense? See, whenever we draw a cell, we will be drawing nicely, like the cell is having this much, like nucleus is there, endoplasmic reticulum is there, Golgi complex. So all these are there when we will be seeing. So let me show the cell diagram also. Wait. See here, when we draw a cell, we are drawing nicely here. You can see nucleus, beautiful nucleus, a big vacuole and endoplasmic reticulum, Golgi complex, and the chloroplast is there here, peroxisomes, mitochondria. So this cell is completely like we are seeing a proper well organized cell here. But whether this cell looks like this when we observe under microscope, it will be having many things. So it will be like, it will be showing what? See, you just think it is a cell now and it will be covered with many. So why they are proper shape of cell is maintained in the sense it will be having many network like structures like this. Many, so many network like structure will be seen upon this. A web like structure which is giving a proper shape to a cell there. So it is a web like structure which is present. This web network like structure is there. No, these are nothing but cytoskeleton. Cyto means what? Cyto means a cell. Skeleton is nothing but it is a network structure which is giving mechanical support to a cell there. So it is actually composed of many things. We have removed all those cytoskeletons and we are showing only the cell organelles here. You can connect to this. See, if we observe a cell here, the cell is there. So a proper nucleus, everything is there. But before that, it is having so many network-like structure like this. So a web-like structure is there, which is giving a proper shape to a cell there. Cytoskeleton. Cyto means what? So here cyto is nothing but cell. Cell skeleton. Cell skeleton is nothing but it is a cell which is giving mechanical support to this one. So when you see a human, so if you just see a human body, when you are studying muscle cells, a proper without a skeleton, you can see that you just type the human body of muscle with the muscles. Only muscles you will be seeing there. Muscular only you will be seeing muscles without any skeleton. If you just type skeleton of a human body, then we will get only skeleton there, isn't it? So it is like this. When you are studying cytoskeleton, we can see only the web-like structures of the proteins there. Cytoskeleton, it is made up of some proteins that we will be seeing in next session. That is microtubule, intermediate filament. I am writing only short form because in later sessions we will be studying about this in depth. Microtubules, intermediate filaments and microfilaments. So these are proteins. So these protein structure which is making a web-like structure there, and this web-like structure, it is making a proper, giving a proper shape to an this cell there. So that's why it is providing a mechanical support, no? So that's why we named it as cytoskeleton. Cytoskeleton structures these are. So based on the diameter, these are of different types: microtubules, intermediate filaments, and small microfilaments there. So it is making a web there. It is about that one. Even locomotory structure. Locomotory is nothing but what? Locomotory, they are talking about flagella, cilia. So we will be seeing flagella and cilia of locomotory structure. Even it is made up of that filaments, protein filaments there also in case of flagella and cilia. So that also in depth we will be studying one by one in the cell organelles topic. So now coming to this eukaryotic cell. So as we discussed, so eukaryotic cell is of two types. Mainly many types are there. The eukaryotic cells we can see in many different type of organism or different group of organism. So when it comes to the plant, so we are only discussing here the plant cell and animal cell differences now. So take a plant cell here and animal cell. So what are the major differences between plant cell and animal cell? So plants also made up of eukaryotic cells, animals also made up of eukaryotic cells. So what difference will be there in between that? Means you need to remember the 4C here, 4C concept. That is, in case of plant cell, the main first thing is cell wall. So cell wall you need to, in case of cell wall, in case of plant, cell wall is present. In case of animal, cell wall is absent. So in case of plant, a central large vacuole will be there. A central large vacuole will be seen. It is present in case of this plant cell, but vacuole usually it will be absent in case of animal cell. And one more thing is, so chloroplast, like plastids. Plastids are present in case of plant cells. It is plant plastids are absent in case of animal cell. Like chloroplast is there, no? Plastids are nothing but chloroplast, chromoplast. So cell wall, central large vacuole, and chloroplast. It is present here. It is absent here. And last one is the centrioles. So here you need to remember, which is very important. Centriole, it is absent in case of this one, but it is present in case of animal cell. Centriole, it is present in case of animal cell, and it is absent in case of this here. So these are the main differences between plant cell and animal cell here. So these are the main differences. You just revise these things and go and revise in your book and solve some DPP's questions in your book, so that you'll get a complete clarity of this concept here. So what we have discussed today, so inclusion bodies, ribosomes, okay, the ribosomes and inclusion bodies we have discussed of prokaryotic cells, and we have discussed about the eukaryotic cells. So related to that, you can solve the questions also, but lack of time. We will discuss that in the next session with the question, and we'll continue in the next session tomorrow. So till that, take care, bye bye, and solve some questions that will help in your revision of the concept. Thank you.